Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Ray Kaplan. He's a professor of infectious disease at University of Georgia. We're going to talk about parasites, roundworms, drug resistance, etc. So, Ray, thanks for coming.
Thank you, Richard.
Yeah. Tell me about your interest. How did you get interested in parasites? How did you run across them?
Yeah, it's kind of an interesting story, and it relates to why it's good to give opportunities to young people. I was, at Virginia Tech as an undergraduate, and I was in a work-study program, a cooperative education program, really, where I would spend 6 months of the year in class, and 6 months of the year I'd be working, and I was working at the U.S. Department of Agriculture. And I went back for one of my work sessions, and due to some budget cuts, they had no funding. So they searched around and found another laboratory that had some funding, and I went to work for them, and it turned out to be a parasitology laboratory. It was just a short 3-month stint there, but it was something different than I had done previously, and I just found it really super interesting. And so then later on, when I went to veterinary school, parasitology was rekindled. My interest in parasitology was rekindled, and so I stayed active, working in the parasitology lab and doing some summer research while I was in veterinary school. And so that really kind of set the stage such that when I decided to leave practice and pursue a career in academics, I decided that pursuing parasitology was the right field for me.
Okay. And yeah, there's a big world inside of every field. So what are you focusing on within parasitology? What kind of organism and what kind of host and what mechanism?
Yeah. So my research now is focused on nematodes, also referred to as roundworms. And this is a very, very large group of organisms that exist as free living forms in the environment. And actually free living nematodes are believed to be the largest group of organisms on the planet, of metazoan organisms, animals on the planet. And then there's plant parasitic nematodes of many, many types. And then there's also parasites of animals and of course humans and veterinary species. So it's a huge, huge group of organisms. So I work in that group, and I focus on parasites of livestock and of dogs, with a focus in drug resistance.
Drug resistance meaning that the parasite itself has acquired resistance, just like bacteria do from antibiotics.
Yeah. Sorry for not explaining that better. So yeah, we use drugs called anthelmintics, which would be akin to antibiotics that are used for bacteria for treating infections with parasitic worms. And there's some amazing drugs, and they've worked really well for decades, but they've really stopped working. And more so, we're seeing more and more resistance to more and more drugs and more parasites, and in some cases to all drugs that we have available. Just like we have multiple drug-resistant bacteria that nothing can kill, we have that now with parasitic worms. So that's the area where I focus my research is to try to understand and solve the problems posed by drug resistance in these worms, where the drugs no longer are working to control them.
Huh. What happens if you have a parasite that usually infects a dog, but I don't know? Well, first of all, okay. So the parasites that would infect dogs, the roundworms, where do they come from? How do they get them?
Okay, well, there's many different types of worms, and they're going to come from different sources. I should say that basically each type of animal has their own worms, and most worms are very specific to a particular host. So a worm that infects dogs mostly is pretty much just going to infect dogs. And cats actually have different species than dogs for most parasites. There is some crossover in closely related species, but mostly they're separate. And for instance, horses and cows have really completely different parasites. There's very, very few that cross over between them. The overwhelming majority are very specific. And even between animals like cows and sheep that are fairly closely related, the majority of species are actually different. They're very closely related. They're like cousins of each other, but they have different species, and only a relatively small number actually cross-infect. So when we're talking about a parasite of dogs, that it really depends upon the parasite. And to give you two examples of parasites that I do a lot of work with in dogs, heartworms, which is probably the most important parasite of dogs. It can really cause life-threatening disease. It's found in dogs all across the country, and it's transmitted by a mosquito. So the mosquito is a necessary part of the life cycle. And so what happens is the mosquito has to bite a dog that's infected with heartworms. During that blood meal, it ingests a stage called microfilaria, which is a larval stage. And then those larvae will develop in the mosquito and go through several stages of development in the mosquito until it reaches the infective stage, that then will be transmitted to a subsequent dog if that same mosquito that now has survived long enough to have those mature infective larvae inside their body. If that mosquito then takes another meal, it can transmit that worm to a new dog. So
what if I don't know if they do, but what if a biting mosquito that's bitten a dog with heartworm then bites me?
Yeah,
puts the heartworm into me. Will I get it or no?
Well, you won't get the heartworm in the true sense like a dog gets it, and you probably nothing will happen. Although in a small percent of cases, humans can become infected, but what happens is the worm, as it'll penetrate into the skin like in a dog, but because it's in the wrong host, it really doesn't know where it's going. It doesn't have any cues, and so it winds up doing some migration. And typically, what happens, it will die during the migration, and a small percent of people will have a small nodule in their lung. Actually, it's completely harmless, but there's a little nodule that can be seen like in an X-ray on a lung. That if it's actually biopsied, will show that it's just a granuloma or a group of inflammatory cells that had encased a small little worm. So it really doesn't cause any disease in humans, although it can infect humans technically and uncommonly, but it can.
It's just amazing that parasites are matched with their hosts and they're so selective to them.
Yeah, no, it is, but it's due to millions or tens or hundreds of millions of years of evolution. These parasites have just adapted to their host. Probably, way back in evolutionary history, a lot of these parasites, as they were basically transitioning from the free living nematodes that weren't parasitic to becoming parasitic, then they probably started to separate out, and they probably were more generalists. But over millions of years of evolution, they became well adapted to particular hosts, and they became so well adapted that then they weren't even able to infect other hosts.
So, looking at the parasites, trying to figure out why they're resistant to drugs, from what I've gleaned talking to other parasitologists, that parasites have their own microbiome. That microbiome probably has phages that prey upon it. I don't know if anyone's found viruses that particularly attack parasites and kill them or make them sick, but they seem to be very amazing creatures.
Yeah, that's an interesting area. That no one has yet found a virus that is a pathogen of a worm that could be used as a mechanism of control. But there's definitely they definitely have a microbiome for sure. But there's really very little known about that. I think that's an area that's going to be ripe for research in the future. But currently, we really don't know very much about it from that standpoint. But what is interesting in terms of a microbiome, now that you bring it up, is that there's been a lot in the news in recent years about how important the microbiome of the gut is to human health, and the same is true, of course, in animals. But what's often overlooked is the fact that while that microbiome in the gut of all these billions and trillions of bacteria in the gut, with many, many different species, some of them are beneficial, some of them are harmful, and so there's a balance, and you want more good ones to remain healthy. That evolutionary process that was occurring in the presence of worms, because worms have also been in the gut throughout all evolutionary times. It's only been in just the last 100 years or so that that people really don't have worms anymore. Of course, animals still have worms in their gut commonly. So the worms actually co-evolved with the host, while the microbiome and what we refer to as the nemobiome, all those nematodes were also in there. So it's really a complex relationship that we're really just beginning to understand. But we do know that we think of worms as being bad things, as being pathogens, and they do cause a lot of serious disease. But there's also evidence that from an evolutionary perspective that worms probably were important for helping to regulate a healthy immune response and prevent hyperinflammatory diseases and autoimmune diseases and things like that we commonly see in the first world. And those diseases are actually quite uncommon in the underdeveloped world, where people are infected with worms commonly.
So there are instances of worms that are actually helpful to their host, or at least not pathogenic. They just live inside them for long periods of time and get along and hang out and do their thing.
Well, actually, that's probably true of most parasites. Really, it's more so numbers. Like, we think of parasites as being harmful, but in low numbers, they probably are not. They're not really very harmful to the host. It's when you get too many worms. And then when I say this, it really depends upon the worm. And this is not an area where I focus and specialize. It's a really interesting area. It's not an area that is my specialty, so I don't want to go too deep into it. But when we see disease in animals from parasites, it's usually because they have too many parasites, not because they have some parasites. And so basically, the whole idea with control of parasites, particularly in a livestock situation, where I really focused most of my research on over the years, is to maintain levels of parasitism in animals below the threshold that cause disease, because it's impossible to eliminate them. We tried to eliminate them by treating them frequently with drugs, and all we've got out of that is that the drugs don't work anymore and the worms are still there. So we're trying to come up with strategies to reduce the amount of drugs that we use and still keep parasite burdens in the animals at low enough thresholds that they're not causing disease. That kind of strategy works well in livestock. It doesn't necessarily work well for dogs for two reasons. One, a worm like heartworm is very pathogenic, and it can cause disease. In just very low numbers of worms can cause serious disease. So you don't want a dog with any heartworms. And another worm I work with, hookworms. It's a blood-sucking worm that lives in the intestine. That's a worm that if there's just a few there, probably wouldn't cause much harm to the dog. But it's one of the worms that actually is zoonotic, which means it can cross over from animals to people. And kind of like the story I told you with heartworm, if a person gets infected with hookworms actually can infect directly through the skin. And so if a dog hookworm infects a human by penetrating the skin, again, it doesn't have the right cues. It can't do its migration that it needs to do. Normally in the dog, what's going to happen is that larvae, after it penetrates the skin of a dog, is going to enter into the bloodstream and go to the lungs, basically penetrate into the lungs from the bloodstream, crawl up the bronchial tree, and then get swallowed back to the intestine. And that route is used by quite a few parasites. However, in a human, when the worm penetrates human skin, it doesn't have the right cues, and so it just starts migrating under the skin, and it causes a very severe hyperinflammatory response that kind of is similar to what you would get from poison ivy, and you can see these tracks under the skin that are very inflammatory and reddened and raised and extremely itchy, just like you would see with poison ivy, and that's what happens when people get infected with dog hookworms, and so that's why at the beach when a lot of the laws for keeping animals cats off the beach, or keeping them leashed and cleaning up after them, that's one of the parasites and one of the diseases humans can get from animals that we need to be concerned about.
Well, instead of, are there worms that will exclude other worms? Are there ones that, aren't too harmful in low numbers, like you said, and when a dog has them, for instance, it'll stop them from getting heartworm or hookworm, or perhaps that's a mechanism by which you can avoid using drugs, deliberately infect them with something that's not so bad.
Yeah, and that's a good thought, but no, that doesn't happen. Basically, each species of worm has its own niche. It's really kind of fairly unique. Even like in the intestine, there's lots of different intestinal worms, but each worm has its preference of where it lives inside the intestine, and so they don't. There's really no competition, exclusionary type of competition. So having one worm won't protect you from another worm.
Are there are there instances of a lot of these worms that will stay in a host for a long time and then only come out, let's say, when the host is under some kind of stress or sickly?
Yeah, that's a good question, because that definitely happens. And I would say the majority of parasites have a finite lifespan that lasts anywhere from a few months to many months, and some can live for a few years. But most of the worms are going to live a matter of months in a host. However, that's kind of once they reach the adult stage. However, there are some parasites, and hookworm is another great example. So that life cycle I told you about, where the worm penetrates the skin of the dog, as a dog in that cycle, where the larva penetrates the lungs and goes up the broncho tree back to the intestine, that occurs mostly in puppies. But then once dogs start getting older and they start developing immunity, what happens is there appears to be like an immune blockade that prevents the larvae from getting into the lungs. So instead of, once it's in a bloodstream, instead of getting having the ability to penetrate into the lungs, it gets flushed away into other tissues like muscle, and then the worms will just penetrate into the tissues around the body, mostly muscle, but many other tissues in the mammary tissue, and they'll kind of go into an arrested state of development, and they can stay in this arrested state, kind of almost like a hibernation type state for years. And then, particularly in dogs, what happens is that after a female dog has puppies and she's lactating, the larvae actually during late pregnancy become reactivated, and then they'll actually travel into the milk, and there's direct transmammary transmission of hookworms from the bitch to the puppies right after they're born.
It's amazing how these parasites can, obviously they anthropomorphize it, they know where to go. Somehow they know they're in the right host. They're reacting to cues. They're able to hang out and hide and monitor the host's, conditions until the time is right. And are there also worms that will go into I don't want to call it a sporulated form, but a dormant or an inaccessible form if the conditions go wrong in a host instead of just dying?
Not really, and they've adapted these abilities to kind of go into this arrested state of development and live inside the host for a long time. Either for like hookworms, it could be for years. So like in most dogs, all those larvae that get encysted will just never come out of encystment. Only two types of dogs will ever come out. It has to be an intact female that then gets pregnant, and then the larvae have the opportunity to come out. And we don't know the exact mechanism, but it appears to be some interaction of the hormonal changes and the immune response. And then there's a small percentage of other dogs we don't know the exact mechanism, but they appear to have some sort of immunoincompetence, even though they're healthy otherwise. And then these dogs, the hookworm larvae can slowly just leak out over time. So these dogs are like, every time they go to the vet, they have hookworms, but the treatment is effective to get rid of them. But as soon as you treat them, more will come back because you have this reservoir in the animal. So we have that. And then in livestock parasites, so the larvae of livestock parasites are going to be just living, in the grass, down at the lower ends of the grass, just near the soil, and then they're ingested while the horse or cow is grazing. And then what happens there is that during times of the year where it's too cold or too hot for the larvae to be able to survive out in the environment, what happens is there's cues that they pick up prior to the adverse time of the year. So let's say like in a cold climate, where of course there's no parasite transmission in the wintertime because the cows aren't grazing in the winter. So the parasites have adapted. Some species have developed the ability to pick up on cues. Again, we don't know exactly what they are, but it appears to have something to do with probably their condition based upon changes in temperature and changes in daylight. And then what happens is when they're ingested on the pasture, instead of doing their normal development as they usually do, they will penetrate into the tissues of the host, of the cow, let's say, and they go into an arrested state for several months. And then they only come out and continue their development several months later. So that would be like in the springtime, when now the environment is conducive to that parasite being able to survive and transmit further. So the parasites do have mechanisms to survive periods of adverse environmental conditions like that. And they can do it depending upon whether it's a cold climate or a hot climate or a dry climate. Whenever there's a time of the year where it's really not going to be possible for that parasite living in the environment outside of the host to survive, and pretty much every parasite has a stage outside of their host. Either has to be in a what we call an intermediate host, like a mosquito or another animal, or it's going to be in the free living environment. But every parasite has that stage in between two different hosts that get infected. And so there's a very, very intimate interaction of parasite and the host and the environment all together. And so parasites have developed mechanisms to kind of overcome some of the adversities that they could see in the environment that would affect their ability to complete their life cycles. But there's no stage like bacteria, that can go into like a spore and just kind of live in the environment potentially for decades or even longer. There's really no form like that for parasites.
But there is a cyst form. Do you think it would be possible to create a drug that wouldn't kill the bacteria but would put them into cyst form, so they go inactive inside a host for a while? That
is possible. The problem with strategies like that is that often it's for many parasites, it's kind of that larval stage when they come out of encystment that is the most pathogenic part of the life cycle. So if you encourage more and more and more to become encysted, when they finally did come out, then you could cause a lot of severe disease.
So how do the current drugs work to get rid of worms, let's say in dogs? And how does the mechanism seem to be failing? Like, how does the resistance manifest itself?
Interestingly enough, we know very little about how these drugs even work, and even less about how resistance develops. Even though we've been studying this for years and years, these worms are super complex. They're much closer, closely related to their hosts, to mammals, than they are to anything like a virus or a bacteria. They have very large genomes. They have a lot of genes. So they have almost as many genes as a mammal does. So these are very, very, and they share a lot of the same metabolic pathways. So these are very complex, higher organisms. And so it's been really difficult to find what's causing resistance to the drugs. For one of our drugs, we have three major families of drugs that we use as anthelmintics. And there's some other drugs, other families of drugs that are used more narrowly. But there's three major broad spectrum classes of drugs that are most often used. And each of those has a different mechanism. One of them we know is it kills by causing paralysis of the worms. Another one is a metabolic poison, and we know it interacts with tubulin and prevents microtubule formation. And microtubules are critical components of cellular structure and just cellular metabolism and movement of molecules through cells. And then what's interesting is probably the most important drug we have, ivermectin, and then the whole class of drugs that we refer to as macrocyclic lactone drugs. We really don't even know how those work. We know we have a target that we figured out. It's a ion channel that we know that this drug affects, and it can be shown very nicely in laboratory experiments. But we know that resistance doesn't occur due to changes in that gene, and there's evidence that there's many other sites of action that we're just starting to learn about. So we really know very little about how these drugs are acting. Another reason why it's so complicated is that we about half the genes that we find in worms, we don't even know what those genes do. And so if these drugs are interacting with some of those genes and pathways that we don't even understand how they work or what they are, then we obviously can't figure out how these drugs are working to kill them or how the worms are overcoming the effects of the drugs. So it's an area that really, even though there's been work for decades, that we know relatively little, and we're learning more and more all the time. And finally, in recent years, with the advancements in genomics, we've been making much better progress. It's kind of one thing that's funny is that I often will get asked by microbiologists who study viruses or bacteria, well, why don't you just sequence the genome of a resistant worm and a sensitive worm and compare them and find out how resistance is what's responsible for resistance. Of course, we've done that many times, but parasitic worms have enormous genetic diversity. These populations are huge. So for instance, on a farm, these worms can produce thousands of eggs per day, and there's thousands of worms infecting a herd of animals. And so literally over the course of a week, you could have 1 billion or more parasite eggs being shed onto the pasture. So this creates incredible genetic diversity. Geneticists estimate that the worms, one species of worm that's a common worm called Haemonchus, that it's a sheep worm, that one flock of sheep, the Haemonchus in one flock of sheep have more genetic diversity than the entire human population. So that what happens is when we compare the genomes of two Haemonchus worms, we'll find hundreds of thousands of differences in sequence. And that doesn't matter if it's two worms, same farm, two worms on different farms, one resistant and one susceptible. It doesn't matter. You're going to see just this huge genetic diversity. And so when you compare them now, if you have hundreds of thousands of genetic differences or SNPs, how are you going to figure out which ones are responsible for the drug resistance? It's virtually impossible to figure it out that way. So
what might be interesting though is because you might be able to use the big data against the parasite. So because it's so prolific, because it's so diverse, you'd probably have to use, again, big data. And I guess I'd put it this way. The parasite is naturally exploring the limits of diversity. If you're able to characterize what those limits are, because you have so many examples, so many samples of a parasite, if you have, it would take a while to sequence them. But if you're somehow able to characterize the range of diversity, it's going to be finite at some point.
Yeah.
And perhaps then you could see trends, clues, conserved regions, et cetera.
Yeah, no, absolutely. That's really, really good thinking there, Richard, because that's exactly what a colleague of mine is doing. Dr. Eric Anderson at Northwestern, he's a geneticist that works on Caenorhabditis elegans, which is a free-living nematode that's used as a model for studying eukaryotic biology and genetics, and also as a model for studying parasite nematodes. And so he's actually been doing that. That's his lab focus on. He's got a project where he's sequenced hundreds of different strains of worms from all around the world, looking for natural diversity. And he's learning an awful lot about how this diversity impacts phenotypes in different ways, including drug susceptibility. So actually, we're working together on a few projects, and so it takes people like that. I mean, I'm a veterinarian, an applied biologist. I'm not a geneticist, and I'm not a genomic scientist. So it's definitely not my specialty. But I'm working with a few other laboratories that do have that expertise, and so we are looking at trying to figure out resistance from that standpoint that you raised, looking at natural diversities that are occurring. But it requires a lot. I said big data, so it requires laboratories and scientists that have the ability to sequence genomes and analyze that massive amounts of data efficiently. And so, like Eric Anderson at Northwestern is one of those labs that can do that. And so, we are working with him on a few projects and have a few more that in the pipeline.
Well, what about comparing within one sheep or one dog the diversity, and then overlaying that on the diversity of a whole, what do you call a herd of sheep, not a flock, a herd, and then selectively treating, let's say, some of the sheep, with drugs, and then again looking at how is that diversity changed within a given sheep, and then does it spread amongst the herd in a different way? I guess if you looked at all those things, perhaps you'd uncover some pathway.
Yeah, I mean, certainly could be possible, but they're the tools are just becoming available to do things like that, and the funding for something like that just probably would be very difficult to find.
The sequencing bill, as I think about it, would probably be astronomical.
Yeah, so when geneticists have looked at the amount of diversity, essentially the amount of diversity on one farm is about the same as it is between two farms. So there's so much diversity, genetic diversity, that any type of project like that would be massive. And so to probably make it work, you need to have a more controlled system. So that's why, like Dr. Anderson is using C. elegans, because it can be manipulated and maintained in the lab very easily, and stored and grown and studied. Whereas with parasites, what becomes very difficult is you have to grow them inside an animal. And so it becomes very, very expensive and difficult to study them, over time, over generations. And so it really impairs the ability to make advances in parasitology, like can be made in virology or bacteriology, where you can grow the organisms in a laboratory. And with parasites, you have to grow them in animals, and they have long life cycles, not hours or days like viruses and bacteria, but literally weeks and months, and sometimes many, many months. Like heartworm's life cycle is over 6 months. So it becomes very expensive to cycle these and on the scale that's necessary to look at a lot of these problems. So it makes it much more difficult, and takes a lot more time to make advancements in parasite research as compared, well, when I say helminth research with worms, as compared to other types of organisms.
So what questions are you, getting close to getting an answer to? Like what kind of breakthrough insights do you think that you're nearing? Well,
we're when I say we, it's not my lab specific. Are you talking my lab specifically, or kind of my colleagues as a group?
Well, your lab specifically, the work that you're doing.
Okay, so right now, one of the areas we're spending a lot of focusing on is drug resistance in hookworms in dogs. So I told you about the ability of hookworms in some dogs to kind of continue to leak out over time. So every time the dog would go to the vet, they'd be positive, and the vet would treat them, and treatment would be effective. But then they'd go back next time, the dog's positive. So we call that larval leak, and that occurs in a small percentage of dogs. Again, we don't really know why, but it's something we observe. And then a few years ago, several of my colleagues and myself started getting calls from veterinarians about these chronic recurrent hookworm infections that they're having trouble clearing. And so most of my colleagues thought that it was just more cases of larval leak, and for some reason larval leak might be becoming more common. But because I've been studying drug resistance, for pretty much 20 years or so, to me it didn't smell like that. So it really seemed like this could be a real case of drug resistance. So we established some strains that we were of hookworm from clinical cases that vets had called us about. We got some of the larvae, and then we infected a few laboratory dogs so we could study it further. And basically, what we found was that the worms were resistant to all three major classes of anthelmintics that I mentioned earlier. And so basically all the drugs that we have that are labeled for use in dogs, none of them work against the hookworms that we're finding. So we've been studying this now for the last couple of years, trying to figure out a little bit more about the biology and the epidemiology and how to diagnose drug resistance and so forth most optimally. And we've been testing some new drugs against it, and so we've recently tested a drug it's sold for dogs in some parts of the world, in Europe particularly. It's not available in the U.S. That's essentially it hasn't been approved for use in the U.S. by the FDA, but we tested it and it worked really well. And we're also now doing a study where we're trying to figure out the geographic distribution and prevalence of these resistant hookworms throughout the country. So we were just getting that going when the COVID started, and then it caused our lab to shut down for the last few months. So it's on a hiatus now, but we should be back up and running soon, where we're collecting hookworm samples from dogs from all around the country. And then we're testing it to see if they have resistance mutations, because we do know for the one group of drugs, the benzimidazole drugs, which are a very, very commonly used anthelmintic, we do know mutations that cause resistance, and we from livestock parasites, and we found the same mutation in hookworms. And so using some deep sequencing assays, some high throughput deep sequencing assays, we're able to quantify the presence of those mutations. So we have a study going on that hopefully will get restarted again here shortly, where we're going to be studying the distribution and prevalence, and at the same time, in collaboration with another lab that does nematode genetics, we're going to be looking at to see if we can figure out the origin of the resistance by comparing the different genetic haplotype backgrounds of the worms that we find. So that's probably one of the major projects we have going on, and really we're the only lab in the world right now that's actually working specifically trying to address this drug-resistant hookworm problem. And then another area I started working on recently was poultry. I'd never really worked with poultry before, but we started. We talked to some veterinarians that were complaining about the treatments not working, and so there's a large roundworm, nematode roundworm of turkeys called Ascaridia dissimilis, and so we did some testing on that, and we confirmed for the first time that there was drug resistance. There had been a few studies that were published in the past where there was suggestions of reduced efficacy, but they really hadn't done a proper controlled trial to demonstrate conclusively that there was resistance. But we actually did a study where everything was controlled very tightly, and so we showed definitively that the worms were resistant. And what's kind of interesting is that the mutations that for resistance that we have found in many other parasites of livestock and in hookworms and dogs, those are all a group of parasites we call strongyle parasites. Then there's another group of parasites called ascarids. They're actually genetically pretty distant phylogenetically, but they're of course they're all nematodes. But so far we haven't been able to find those mutations, it's still not definitive that they're not there. We're doing some more sequencing, but we haven't found them yet, suggesting that there may be another mechanism involved. So that's an area that we're really interested in pursuing. And again, that's an area that I'm working with Dr. Anderson on to try to understand the other diversity of mechanisms of resistance that might be out there.
So are parasites if you compare them to bacteria or viruses, they seem to be able to proliferate and mutate and change faster or just as fast?
No, it's much slower actually, because one big difference between viruses and bacteria and parasites, and again, the helminth, the worm parasites, is that viruses and bacteria, they reproduce clonally. So by asexual reproduction. So you have one virus and it just or one bacteria and it just creates billions of more of the same. With parasites, each parasite has essentially its own genetics, and then they reproduce sexually. So there's a male and female. So you have to have a male and female mating, and then once that happens, then that female can produce a lot of eggs, which can then get into the environment or be picked up by an intermediate host and infect other animals. But because it requires sexual reproduction, that changes the whole dynamic of occur and how rapidly those changes are going to occur. So you can get a lot of genetic diversity because a female will produce a lot of eggs. And so each one of those
can have where in a field of sheep, where is the sexual reproduction happening? Inside the sheep?
Inside the sheep, yeah.
Okay, and then they're pooping out, I guess, and then it's picked up again, it's recycled into another sheep?
Yeah, so what happens with these worms is that you have the larvae in the intermediate stages or out in the environment that will then be ingested or penetrate the skin. And they make their way into whatever niche that worm lives. Most commonly, it's in somewhere in the gastrointestinal tract, but it can be almost any tissue in the body. And then you'll have some worms that develop into males, some develop into females, and then as once eventually they become sexually mature, which again takes weeks or months depending upon the worm, then they'll mate, and then that female will start then shedding egg stages or larval stages that can be available to infect other animals. But the large majority of those, of course, the chances that any one of those eggs or larvae is ever going to infect another animal is super low. So the way the parasite deals with that is just by creating a very, very large number of those eggs and larvae. And so only a fraction ever will infect another host, but because they produce so many that they basically ensure that the life cycle will continue.
Well, within a sheep, for instance, are there more males versus females? Is there a lot more diversity in the females versus the males? Like, are there any skews that you can capitalize on? Any bottlenecks that, in terms of resistance? Has it been discovered that males are more resistant than females, or vice versa?
Mostly not. There has been, in some species, there has been found some sex linkage in some drugs to resistance. But for the most part, there doesn't appear to be much of a difference between the susceptibility of males and females to the drugs. And you might think that you don't need as many males as females because actually they're polyandrous. So they'll mate with multiple partners. But in reality is that the percentage of males and females is often pretty close to 50-50. So it's an interesting thought, but there's no evidence that there's that much the differences there. Although there are sex chromosomes in worms, just like in mammals. But again, that's not an area that I have expertise in, so I don't want to go too deep into that. But it's an interesting thought. But so far there's nothing that's quite very apparent to suggest that looking at males versus females is something where that would be highly fruitful for most things in this area.
Yeah, I don't know if that would be a good thing in a cocktail party, to say that you're an expert on parasite sex. that would probably turn people off quickly. So, well, very good. I mean, it seems really interesting. The stuff you're working on. It's got its own really difficult challenges, and very, very interesting. What's your guess or estimation on what you think you're going to figure out in the next few years? Is it just who knows, or do you feel like you're close to understanding something?
Yeah, well, one thing that it's not something I'm directly working in, but I'm collaborating with some other labs, genetics labs, and they are coming very close to figuring out the genomic loci responsible for ivermectin resistance. So ivermectin, again, probably the most important anthelmintic drug. The discoverers of that drug won the Nobel Prize a few years ago, Nobel Prize for Medicine. It's used in humans. Billions of doses have been delivered to humans. Billions of doses have been delivered to animals over the last few decades. It's really a miracle drug in many respects, and it's done amazing things for human and animal health. But we've had big problems with drug resistance in the last couple of decades, and it's getting worse and worse. But we still don't know. We have absolutely no knowledge of what causes resistance. But they have identified using some of the more newer genomic tools and then some clever strategies. They have actually figured out where on the chromosomes. There's one chromosome in particular, and the loci on that chromosome, where the region of that chromosome where the resistance mutation lies. There's still a fair number of genes there, but they're narrowing it down, and within a few years, I believe that we'll actually know the primary loci that causes resistance, ivermectin resistance. So that's going to be a huge breakthrough. We don't know if that's going to be the only one, but it certainly is going to be the major one. So that's one major breakthrough, and for sure. And then as far as the things, it's more, I say, incremental. The work we're doing with the Ascaridia could be very interesting because it's Ascaris, which is a closely related worm in the same group as Ascaridia in turkeys that we're working on. Ascaris lumbricoides is the most prevalent parasite of humans, helminth parasite of humans. There's about 1 billion people around the world infected with Ascaris. So it's an important problem in people, and especially children. So, and a lot of drugs are used to treat it. The same drugs that we use, similar drugs that we use in turkeys. And so if we can understand how resistance is developing and figure out the mutations responsible for resistance in Ascaridia, that could have important direct application to surveillance of resistance developing in the human Ascaris.
Well, very good, Ray. It's been a good call. We're out of time, but what's the best way people could find out more about your particular work in your lab?
Yeah, it's a good question. I wish we had a better website. I have my lab, the department website that has some general information, but it doesn't really go into a lot of detail. As far as the sheep and goat work that we do, I didn't really talk all that much about that. That's more applied. But I'm involved with a large group of scientists of all different specialties, extension specialists and nutritionists and animal scientists and agronomists, and veterinarians, parasitologists. We all work together, and we have a website called wormx.info. So it has basically lots of information on controlling worms in sheep and goats. And so that's an area that anyone who is interested in that topic can go to. As far as the other things I talked about, I don't really have there's no good website that has a lot of information, unfortunately, right now for that.
All right, but that's a good start. I just want to give listeners, somewhere else to look. So, very good. Well, Ray, thanks for coming on. I appreciate it.
Yeah, thank you, Richard. It was interesting talking with you, and you had lots of good questions, and I enjoyed speaking with you today.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.